Wow, I can't believe I'm here. This is just so exciting. You have no idea. I am originally from Quebec, uh, born in Sept-Îles, Quebec, and I lived 3 years in Quebec, and Quebec City is my most favorite place in the world. So the chance to come, I haven't been back in so long, and the chance to come back is so just, I'm just so delighted. So thank you very much to Juan Carlos for inviting me. Can you hear me? Yes? Okay. Let me know if I need to talk a little louder. They have me all here. I feel like a robot or a rock star, you know? So thank you for coming today, because what I want to talk to you about is about herbarium-based research, research that takes advantage of herbarium collections. And I want to just talk to you through sort of the lens of what I do, how herbaria have been very important to the research that I do. And so I want to take you through a few vignettes of that. But first, there are some of you, I'm sure, here who perhaps don't even know what a herbarium is. And so I want that's possible. And so I just want to explain it. Many people explain it as being a library or a museum of dried plant specimens. That's one way to describe it. And early herbalists in the Renaissance period discovered that if you collect a plant and you dry it and you fix it on paper, that it can, it's an excellent way of preserving them for study. And we know now, after hundreds of years, that these plants do keep their salient features for a great long time. And so it's a wonderful resource. And the first herbarium is attributed to this Italian physician, botanists. Most physicians at that time in the late Renaissance were also botanists. And this was Luca Ghini. And he used to take his medical students around a botanical garden for walks and show them plants of medicinal use and so on. And that was fine in the summer, but in the wintertime, it wasn't so great. So he came up with the idea of creating these herbals. So they would dry plants, just like many of you probably did as a kid, take a special plant and put it in a telephone book, and then he created these herbals. And so these books that were just all these dried plants. And it took off and the idea, the art of making herbaria was disseminated all over Europe by the 1500s. 200 years later, in 1700, the Swedish Carl Linnaeus, he was the founder of our botanical, or all scientific binomial system of scientific nomenclature. And he came up with the idea of, instead of binding these specimens all in books, that we put them on separate sheets, and we put the sheets in cabinets. In those days, they were wood cabinets. Today, they're steel cabinets mostly. And they're classified according to some system so that they are easy to retrieve. So a herbarium specimen is depicted here on the upper left, and it consists really of just two elements, the plant and the label. And the label is usually affixed to the lower right corner. And a plant without a label is no good, and a label with no plant is no good. So they are permanently tied together. And when you have the plant, this is an example of a plant that was collected in Costa Rica. It has the identification of the plant, information about who collected it, when it was collected, and information about the locality, that sort of thing. And so these are very integral parts. And when you think about, people have been making herbaria and contributing plants to herbaria for over 4 centuries. It's like a magic window, a magic carpet to go back in time. And it's our best view of how Earth's vegetation has changed through time. And let me tell you, there are a lot of herbaria, more than 3,000 herbaria in the world. Even Greenland has a herbarium. And it's estimated that about 500 million plant specimens are dried and stored in these various herbaria across the world, some smaller and some larger than others. And I think I have it close in the geography, but of course, here you have the wonderful Louis Marie Herbarium. And it is, at Laval, it is certainly one of the largest herbaria in Quebec and maybe Canada. And it's a very, very valuable and important collection. And so if you haven't enjoyed or had a tour of the herbarium, I recommend that you check it out. I also work at Duke. We have a lemur center. And I tell you, it's very hard people go to the lemur center to see the lemurs all the time. Nobody wants to come and see dead plants in these cabinets. But once you look at them under a microscope and get to study them, the whole world can open up for you. So until recently, the world's herbarium specimens were pretty much under lock and key. If you didn't know somebody, if you didn't know where it was, they were basically hidden from view. So there are herbaria in museums. Almost all universities have some small or some large herbarium, and students often do their degrees there and never even have a clue that resource is there. And it's been accessible really to, mostly little old white men and to very few people in the field. So what has happened recently is that people have begun to take pictures of these resources, not just herbarium collections, but also all kinds of museum collections. And at first, this was done a little bit slowly, where a plant specimen would be placed on a flatbed and has a high-resolution camera, and it's well lit, and you can take pictures of these specimens, very high-resolution images of these specimens, and then these images can be placed online, and people anywhere in the world can view at a high resolution any specimen they want that has been digitized. So of course, doing it this way is rather slow. I saw in your herbarium you have a wonderful guy who was sitting there doing this very patiently. I thought one could only do about 100 a day, but he said yesterday he did 300. So that's very impressive. It takes a very patient person to do that. And but now it's become almost an industry, and especially in the US, where there are some companies now that will come into, for example, at the Smithsonian Institution, this company comes in, these people just go through and digitize all these specimens on a conveyor belt system so they can process 4,000 to 5,000 specimens a day. So it's getting all of these specimens that have been tucked away in the dark, invisible, it's bringing them out into the light and available for people to study. And I'm not really familiar with how progressed or advanced digitization is in Canada. I know it's starting, but in the US, for the last 10, 15 years, there has been dedicated portals that have been funded by the National Science Foundation, like iDigBio, where they have amassed digitized data from plant specimens, mollusks, birds, butterflies, any museum collections. And so these are very competitive grants, but people apply for them, and often as collections of herbaria across the country, maybe 30 herbaria. And we've had a few of these at Duke where we have done, for example, the flora of the Southeast. So everything that's collected from the flora of Southeast, and I think we have about 150,000 specimens. All of them have been digitized, and all of them are available on these portals that you can go and check. So, for example, in my area, a portal I check is the Southeast Regional Network, known as CERNEC. And I can just type in a name and press the list display button, and whoops, it comes up with specimens everywhere. These are from Alabama. Any herbarium that has had its specimens digitized, I'm able to look at them. This is just a short list. It scrolls for pages and pages and pages. And also there's a little thumbnail if there is a high resolution image of that, and I can click on that and I can look at the details of the plant. And it's been a real help in that, you know, for centuries, people have borrowed specimens the same way they borrow books. They borrow on loan. So frequently we send specimens all over the place, and so does the herbarium here to different scientists studying things. But people are now able to look at these high-resolution images, and sometimes they get the information they need just from that and don't need to borrow the specimen, but sometimes also they do need to see the specimen. In fact, when digitization became such a popular thing in the US, I remember one of the deans coming to me and saying, "Oh, it's fabulous," because our herbarium is as large as yours here at Laval. It's about 800,000. So it takes up a lot of space. It's a lot of steel cabinets in a room. And so they'd love to get rid of it because they think, oh, you know, this space could go to a neurobiologist who would bring so much money and everything. It's such a waste of space. You just have dead plants and dead space. And he said, so it's great when it's all digitized, we can get rid of it, eh? And I said, no, because it's not like a library, not like books. We are able to get DNA out of these plants, even 100-year-old or more specimens. And so, no, nobody's throwing away these specimens. And his face went, you know, well, and you have to keep on people to remind them. And that's sort of the essence of what I want to, the message I want to give is how I have used them in various ways. But I must say, I was delighted to look on the portal here at the Louis Marie Herbarium, and I found this specimen. It was collected by yours truly when I was an undergraduate at McGill in Montreal, and yeah, it was back in the Cretaceous, and but it was a project I did working on Mont Rigaud, which is outside of Montreal, and it's interesting. I look at that, and it's a lycopod, and it's a part of the group of plants that I work on. So it was sort of a little signal. I had no idea then that I was going to focus on those plants. So. I've never done this before, but I would like to dedicate this talk in a way to this person, Vicki Funk. She's a senior research botanist at the Smithsonian Institution. Vicki was a very remarkable person. We lost her. She passed away last week from very aggressive breast cancer. And it was a shock really to the community because she was a powerhouse. She was also head of the biological diversity of the Guyana's program and an expert on Asteraceae. So she published many, many books and was very influential. But she was also an ambassador for herbaria. And I thought with her passing just last week, I remembered she published a paper in 2004, a little paper where she decided to call it 100 Uses for a Herbarium. And she wrote it down and she came up with 72, well, at least 72. And then she encouraged the readers to, you know, you can add to the list. But that was very typical of Vicki to be a strong advocate for herbaria. And so what I'm going to do is I'm going to go through all 72 examples. No, I'm just kidding. Just 4 examples. And so the first one, and this actually was the first one on her list, was that a herbarium, specimen has uses in just basic functions and research. And one is you can discover a plant that is new to science in a herbarium. And this really came to note, I think, with this important paper that was published in PNAS, I think it was in 2010. And the title is Herbaria are a major frontier for species discovery. And they, based on their study that they did, have shown that herbaria are unbelievable reservoirs of undescribed diversity. And I kid you not, it happens almost regularly that things that have been collected a long time ago are rediscovered in these dark closets or on these portals and are described as new species. And so their message was that in this particular group, angiosperms they were working on, the estimated 70,000 species are still to be described, and probably half of them are already collected and in herbaria. And so that they were encouraging that effort and research focus should also go towards herbarium research and the field, because it's getting harder and harder to get permits to do research in the field, and field work is awesome, but you can't always go everywhere when you're interested in a particular group. And it's too easily forgotten that this is an incredible resource that's there and just waiting. So as I hinted at, these are my babies. So Juan Carlos is down at this end here with his bryophytes. And, you know, all the people who work on flowering plants and seed plants, well, they're much more important than us. They're over there. And I'm on this little trajectory in the middle. I love ferns and lycophytes. And so that's where I have focused a lot of my energy. And one fern that you might all know is this beautiful adiantum. How many have you seen this lovely fern in the woods locally, Adiantum pedatum? And I happen to have the good fortune of having this researcher in my lab, Dr. Lane Hewitt, who's an expert on adiantum worldwide. And I must say, when I started working with her, all I really knew was adiantum, oh, the beautiful maidenhair fern. And everybody knows that. But what I didn't realize was that it had such a diversity of leaf forms. Maybe I'll take this. So not only do you have those very dissected leaves, but you have adiantums that have simple reniform leaves, and adiantums that have these cordate or hastate-shaped simple leaves. Some that are once pinnate, and some that are just like pedatum have this very complex leaf structure. And the reproductive structures are located on the edges of the leaves, on the backside of the leaves, and they can either be one long continuous row or several little discontinuous rows. There's a lot of variation, and so Lane and I worked for about 10 years, and we produced a phylogeny, and the purpose of our phylogeny was to be able to understand the evolution of the leaf blade structure. So this was our phylogeny, but I want to say that while we were doing this, we were borrowing specimens from everywhere. And there was a specimen that landed on the table from Berkeley that Lane looked at and said, collected in 1894 from Shasta County in Northern California. And sure enough, we did some DNA work and morphological work, and we were able to publish a new species, Adiantum shastense. It's pretty unbelievable. Shasta County in Northern California has been very well explored, that we can still find species there and describe them 121 years later is pretty remarkable. So it speaks a lot to that. And this nice little fern joins a nice bunch of other creatures, the Shasta County salamander and some flowering plants that are endemic to that particular region. So that's just one example. There have been other examples where it's just one of those very exciting moments when you find something and you know it's going to be new to science. So a herbarium also has the ability to provide you material where you can make detailed morphological measurements. And as I've alluded to before, you can take small pieces of the specimen and use them in DNA analyses. And that's been done for about the last 20 years. 30 years ago, nobody even thought that was possible. And so it's been quite a revolution, because all the techniques, molecular techniques that were invented or involved in the medical industry have been repurposed for doing the kind of research that we do. So I don't know if you get this, but people feel sorry sometimes. Oh, you don't study flowering plants, you have no seeds, you have no flowers, you have no fruits. And we have this theme, no problem. Adventures in fern biology are out there. You don't need all those things. And what I'm going to do now is a painful exercise, and that's going to remind you, you may not all be botanists, I don't know, but go over the life cycle of a fern. I hope you've all seen ferns out in the woods, and I just want to do this sort of classroom exercise. I am one of those nerds that loves life cycles. Everything you want to know is in the life cycle. But most people, students usually hate them. But this here is your typical fern out in the woods, and it's a diploid sporophyte. And it's a sporophyte because it produces spores in sporangia. And these little packets are where meiosis happens and where you go from diploid to haploid spores. These little spores jump away from the parent plant, find a nice little place in the soil, and germinate into what are these beautiful little Valentine Day. They're actually little hearts, many of them. And these are called the gametophytes because they produce gametes, male gametes, the sperm, and female gametes, the egg. Sperm and egg get together, produce a zygote, and you end up with a new sporophyte. Now this is, all plants do this. All plants have this alternation of generations, but only ferns have separate. Even your mosses don't do this. They're always, they're always together. So it's incredible that you can, these two parts of the life cycle are completely independent of one another. And we don't see this in any other plant group except for ferns. And that allows us to think about a lot of interesting questions, especially with regard to breeding systems. So when we people collect ferns, they don't collect gametophytes. They're too tiny, they're the size of your fingernail. But what we're able to do is to poke around, remove sporangia from the back of the leaves, sow the sporangia on some media in a Petri plate, and we can grow up living plants of the gametophyte phase. This is a male gametophyte, and this is a female gametophyte of this particular thing. But we can sow spores from a herbarium sheet, and within 10 days, we go from a dead plant to having all kinds of wonderful little babies that you can do all kinds of experiments with. And so that has been, I think, a real advantage to studying this group of organisms. Now, the next slide, don't worry, it's a little busy. But I want you to focus really on the gametophyte, because in textbooks, when you look at a life cycle, it's always presented in this way. So here's the sporophyte, here's the gametophyte, and it's always presented in the way that a sperm from this gametophyte fertilizes an egg on that gametophyte, and you get a new sporophyte. And if that was the only way ferns did it, they'd be the genetic consequences of that would be very, very grave, because that results in having individuals that are 100% homozygous. So it's not great for variability. But ferns don't just do that. They are capable of gametophytic selfing. They also do sporophytic selfing, which is like selfing in flowering plants, where gametophytes from different sporangia, but on the same sporophyte, sperm meets egg and you get a new sporophyte. And they're also capable of outcrossing. So two completely different individuals release spores, and a sperm from one meets the other, and so on. So we have these three types of sexual reproductive things that can happen, or breeding systems: gametophytic selfing, sporophytic selfing, and outcrossing. And this terrible little diagram, I'm sorry, is just to show you that we can know by looking at a dried herbarium specimen and taking off a sporangium and counting the number of spores in a sporangium, if it is a sexual fern, if it is a fern that practices going through sexual reproduction, it will always have 64 spores exactly. And so the meiosis, the spore mother cell here that undergoes meiosis, goes through some 4 rounds of mitotic divisions to have these 16 sort of diploid cells that go into meiosis, and you do the math and you come out with 64 haploid spores. And so that is something that we're able to get information very easily by zooming in on the sporangia. But ferns also do something a little strange. They do something called apomixis. And about 10 to 15% of ferns, it's estimated, are apomictic. And that is an asexual lifestyle where the spore mother cells inside of the sporangia undergo a special event just before meiosis. It's called a pre-meiotic endomitosis. And you wind up at the end having 32 spores. So you can always identify asexual ferns by counting the number of spores. And they're usually larger because these are double the ploidy that go into meiosis. So this is very exciting to me because I like to work on, well, desert ferns. And I work with this person too. So the arrow is pointing. I've worked with Michael Wyndham for about more than 20 years on desert ferns. People think, desert ferns? You know, you must go to Panama and all these wonderful places. And it's, yes, that's what people typically think of as a wonderful fern habitat. But this is Oregon Pipe Monument, National Monument in Arizona. And Arizona has got more ferns in it than Hawaii. But you have to work to find them. And they grow in very tough places. It's some of the toughest field work I've had to do, climbing and hanging from rocks, because these ferns just grow literally out of the cracks of these rocks. So it's pretty bruising field work, especially if you land on a cactus. And I've done that too, so. And these ferns that grow in these habitats, and they grow in these habitats all around the world, dry xeric environments, and they often cope with those environments by having hairs that are densely hairy or furry, or having leaves that on the undersurface have a waxy farina that helps to reflect sunlight and allows them to cope, because they are often 100% always in direct sunlight. And the genus Pellia, which has segmented its leaves into tiny little waxy segments. So they have all these sort of morphological ways of coping with the xeric environment. But we can take our herbarium specimen. There are so many collections of chylanthoid ferns already. We can take a sporangium off with a little razor blade, open it up, count. Do we have 32 or do we have 64? So right away we know something about the sexual breeding system, whether it's an apomict or asexual. And also we can then measure the spores. And the spores have been shown to be correlated with polyploidy. And so we are able to find things just by measuring spores, and it's something you have to train, but you get really patient undergraduates and you force them to do this. And then they think after a month, oh, it's not so bad. But they can find new species, and they have, just by doing measurements. There are some taxa that we only know as, of these desert ferns, that we only know as triploids. There had to be a diploid parent. And all you have to do is measure the spores. And I have a PhD student now working on fern in Australia. And she went through 600 specimens before she found the diploid. Whew, the roof blew off the house, I tell you, when she found it. But it's, you know, it's an incredible thing that you can take these spores, count them, measure them. You can, if you can grow them up enough, you can get chromosome counts, and chromosome counts have been, because ferns are riddled with polyploidy and hybridization. And so you can see here, for example, this is a haploid spore, and this is a diploid spore, and that their size does correspond to carrying a greater DNA content. So by having this information that you can take off of a herbarium specimen, and then you can just take a sample of the DNA, we've had people that have, on various groups that they worked on, double the diversity. So this, for example, was a small project in a graduate student, Amanda's project on Myriopteris. And going into this, we only knew the taxa that were in black. By doing these spore measurements, these spore counts, chromosome counts, and DNA, she was able to fix the puzzle and find critical missing diploids even, and apomictic members. So they literally cross back and forth, and it can be very confusing, and they can all look the same, but we have our characters and they work, and but it requires patience. So they say 10, 12,000 species of ferns. I'm gunning for 25,000. We're going to beat them all. Okay. Third one. We still have 69 to go. Is that a herbarium is a great place to have, to reach out to the public, to reach out to anybody. And to serve as an educational tool for the public. When people find out about it, they're very interested, and it seems very sad to me that we have this profession where we do these exciting things and people don't know about it. So I like to share that. And so this group of ferns that I work on with Michael Wyndham, these are known as the chylanthoid ferns, these desert ferns. There are about 500 species worldwide. And again, for about 15 years, we've been working on this group. And it was a mess, a mess. Things people would call anything chylanthes if they didn't know what it was. It was basically like a garbage can. And so with using these techniques of spores, spore measurements, and DNA, we were able to, this is a sort of a summary of the phylogeny of these 500 taxa, but we were able to, you know, figure out who's who and who needed a new name. And I had a student who came to the lab in 2010. His name was Fei Wei Li. He came from Taiwan. Super guy, knows his ferns, Asiatic ferns, incredibly. And when he arrived, he said, I want to work on something North American. I want to be an expert on something that's strictly here. And so we said, well, Fei Wei, and I've just blown up one thing, but because true chylanthes, because we know this because of typed specimens, is in this clade here. Everything else that's been called chylanthes needs a new name. We can't keep calling it chylanthes. It's not chylanthes. So we had new genus number 2, new genus number 3, new genus number 4, number 5. And we said, hey, Fei Wei, why don't you take new genus number 2? You work a little project, because Fei Wei wanted to work on the evolution of photoreceptors, but he's a wonderful field guy, not just a lab person. And so he said, great. And we sent him off into the field. Oh, you can tell the guy is a very energetic person, loved finding these plants in the field. And if he had only been able to do molecular work based on, this is him in Costa Rica doing field work, his final study, so we had only a few samples of genus number 2, but he was able to, get these samples from the field. But because we can extract DNA from herbarium material, he was, able to more than double the sampling and get a much better picture of this fern genus number 2 that is restricted to Mexico and Central America. And so that's why I have you can do field work and you can do both, and it's a plus magic situation. So while we were working in the lab, it's very exciting to have dynamic people like that in the lab. In the real world, they were listening to things like Lady Gaga in the lab. What's this Lady Gaga stuff? And I sat down for the Emmys in 2010, and I saw this. So I don't know about you, but I was like, oh my God, she's speaking to us. I fell in love with this singer, and I truly did. As a person, she's fantastic. She advocates, somebody of her now celebrity and wealth, advocates for bullies and poor, and that sort of thing, people, disadvantaged people. But when I saw her come out in this costume, and at the time, they were also saying things in the media, I don't know if you remember, but they were saying things like, she's too smart to be a woman. She's bisexual. Very bizarre things like this. And this is a bisexual gametophyte. And so there was one thing after another that just said, we have to call fern genus number 2 Gaga. And so the LGBT community contacted us. We were, because they didn't even know ferns have sex. And that plants have sex. It was fascinating to be able to talk with people. But it got even worse when we did an alignment, of all the DNA. And I kid you not, there's a molecular signature in all 19 Gaga species and not in the closest relatives. So it was in the DNA. What choice did we have but to call it Gaga? And so some people gave me some flack for that and said, "Oh, it's a shame you did that. You should have named it after somebody who, has worked on ferns for a long time and earned it." But I think that the coincidence of everything and the opportunity to be able to tell the world about kailanthoid ferns. I mean, who cares about us working on these desert ferns, right? But you work on them and you love them, and it's a way to touch with culture because we're funded by government agencies. Taxpayers deserve to know what we're doing, and we don't owe it to people to be just in our little white lab coats and saying, "Oh, you wish you were like me," It's making it accessible to them. And so it was a great delight when Lady Gaga's team got a hold of us. When she released her Born This Way album in 2011, she opened her concert in Costa Rica, and she knew about the fern, and because we had to ask her permission. So I have not an email directly from Lady Gaga, from her manager saying, go for it. And she hired some botanists and they went out in the field. And so this is a fern a species that we named after her family, Germinata. So her real name is Stephanie Germinata. So this is Gaga Germinata. And of course, she even put it in a Versace vase and sent a picture. So that was great. But if in 2012, when we published this, if you had put these words into a Google search, like Gaga fern, you wouldn't get any connection. Now you get, almost 2 million hits. And the story, if you want to explore it anymore, I welcome you to just put in Gaga and fern, and you'll see more than enough about this entire story. But the thing that was so interesting was how it piqued people's interest. And so we had people from Rolling Stone magazine, TMZ. I mean, they only talked to rock stars. You know, here we were suddenly very popular, but talking about herbarium and ferns and sex. And it's all turned out to be a marvelous hot conundrum of things that everybody, so it's been a good thing, I think, and I am delighted that we did it. Fei Wei was shocked initially, but he's such a good guy that he took it very well. So my fourth story. So herbarium, and this wasn't on Vicky's list, it can be used to investigate long-term interactions between plants and other organisms. So if you look at this herbarium specimen of Caria, what do you notice? It's full of holes, right? And those holes were made by insects that were feeding on the plant before it was collected, pressed, and mounted and put in the herbarium. And herbarium specimens preserve signatures of their interactions with insects over time. Now, they only go back 400 years, but you're able to not only look at the plant, identify the plant, but you're able to get some knowledge because you also have that data on the label about where it's collected and when it was collected. And so you have information where you can link, oh, those insects were eating there and at such a time, and you can go back through climate records. So they're really unmatched repositories for these sort of insect patterns because, as you know, insects and plants have had a very long history, over 400 million years. It's been a co-evolutionary arms race. And Ehrlich and Raven, the two famous biologists in '64, hypothesized that herbivorous insects drove the evolution of plants, and then plants in return, their adaptations and defenses to insect attacks have stimulated the diversification of insects. So these are two of the largest, most biodiverse lineages, plants and insects, and they've been going at it for so long. So people are very interested in these sorts of evolutionary and ecological interactions, but to study them in the present, a lot of people study them. There are a lot of studies, the study count on this axis, but it's hard work to do. And so a lot of these studies covers, 1 to 3 years, maybe 5, maybe 9 years. It's very difficult to accumulate data sets that cover a long span of time. And so a year ago, I met this woman. This is Emily Meineke. She was a postdoc at Harvard for 2 years. She's an entomologist, an insect ecologist, and she came up with the idea to look at herbarium specimens to investigate insect damage patterns through time. And she sort of came out of nowhere. I'd never heard of anybody doing anything like that. And I was very excited. She'd moved back to Raleigh, and she's been a postdoc in my lab for the past year. She has a job now. She's going to California, Davis. But she has written and been an editor on several very important issues of the unrealized potential of herbaria for global change biology. Museum specimens provide novel insights into changing plant-herbivore interactions. And the one study, she realized you look at these specimens and you see signatures of the damage. You can see serpentine mines, you can see interior leaf holes that show leaf feeding, skeletonization patterns that are left by certain insects, and margin feeding. There are all kinds of patterns. And so she studied these patterns for 2 years all by herself under a microscope with she'd take herbarium specimens, she'd put this grid that she made, and she would, physically count the number of, she did it in a very randomized way, to count insect damage. And the long and short of it is, as she was able to do this for 2 years, 600 specimens, she looked at 4 different woody angiosperm taxa. So Quercus bicolor, Vaccinium, the blueberry, Caria, and Desmodium. So these are 4 angiosperm plants. And because she was working at the Harvard Herbaria, she only looked at Harvard specimens. But she was interested in this claim that New England especially has had winter temperatures that have increased to a great degree. And by doing her study, she was able to show, because she looked at specimens that dated from the 1890s until 2012, I think, and she was able to show that herbivory has been remarkably, it has increased over time in that last 100 years by 23%. And so she was able to use climate data and hypothesize that with global climate change and increasing temperatures, these insects have a longer period of time where they can munch on insects and do their damage. So that was a great paper, and I said to her, well, wow, we should work together. It'd be very exciting. I've never thought of that possibility. And so we said, yeah, and especially now, all these specimens that are digitized, because these 4 species don't just occur in New England, they have very broad ranges through Canada and the US. And so what she was able to look at by hand, this is the years collected, latitude, longitude, and the span of winter temperatures. By looking at digitized specimens, we would be able to really increase how many years we could cover, the space we could cover, and so on. And so these are various digitization projects that are ongoing. There are many, but these are many institutions that are digitizing their specimens, and they're in the millions. So it's big data, if I've ever heard of big data. This is incredible resource that we have online, but we can't have poor Emily sitting there doing it by herself. And we thought, oh, wouldn't it be cool if we could get a computer to do it? And so machine learning is all hot now. And why not, if we could take all of this data, it's all available in high resolution, and annotate it and train computer algorithms to be able to identify insect damage accurately, then we would be able to tell even bigger stories, and we'd be allowing other people to ask their own stories about these plants that are available. So I have a colleague in the computer science department, this wonderful Italian, Carlo Tomasi, who is very involved in imaging and facial recognition science. And for example, in dermatology now, people, when you go and you have a mole that's scanned, and it's run through a computer algorithm to determine if it's a bad or cancerous or not, just because it's been trained by so many. So he's worked in medical imaging, and he thought, whoa, this is a great plan. So I was so excited to get him on board. And pretty soon he had us using this annotator. So we can download all of these images that are online and then call them up into this annotator and then start the process of annotating for different types of damage. Well, that's when we got to work, because you have to have a human part before you can have the computer part. And so this annotator allowed us to put however big, large, because you can't just say, here's a specimen, tell us what kind of it, you have to train it. And so it needs an expert to go in and say, this is interior feeding, this is margin feeding. And we did about 10 different types of insect damage. And we did this on as a preliminary study, because to do this, we're going to need some funding because it takes people time. And so we were able to do this, and on our small data set, I did this on Onoclea, but our main data set was this Quercus, where we identified these different instances of damage, so margin feeding, interior feeding, skeletonization, and so on. And so we have annotated all of these various sheets with this information. It's super preliminary data because this kind of computer algorithm is extremely data hungry and needs a lot of data. And so he was able to take our preliminary data set and divide it into a training set and a testing data set. And then it goes into this magic black box that these computer scientists do, where he's training the classifier to determine what type of damage it was, and to go back and forth and back and forth. And then when we have the algorithm, he's able to use testing data that the computer hasn't seen, and to determine how well we're doing. And this will take a very long time. It will take hundreds, tens of thousands of human annotations. But once that's done, the computer, we hope, will be able to do it. And in our little preliminary study, we show here the squares that are on dashed lines, for example, are things that were done by a human, and the solid lines are the computer. And our model was able to return the correct answer 80% of the time. That's not bad for just starting out. And there's so many other features that can be used to really make this model better, looking at color, looking at texture, but you need the images, and you need the computer expertise. But he was able to produce this, what's called, I think it's great, a confusion matrix. And what you want is for all the non-zero numbers to be on the diagonal. So this is what the human annotator identified as margin feeding, and the computer 114 times also identified it as margin feeding. But you see, we're still in the beginning. 39 of the times the computer identified what we said was normal margin as margin feeding. But the statistics are good. Carlos we put in a grant to the National Science Foundation, and so we're keeping our fingers crossed and hoping that this will be sort of our next. It's exciting. You can, again, all these images, who would have thought that you get, people, 400 years ago, people going out in the field and collecting specimens and then amassing them in these herbarium collections. And 20-some years ago, when we discovered, wow, we can get DNA and learn something about the biology of them. And now scanning, producing these high-resolution images. And just last week, Google was at a biodiversity conference, and they have initiated this white paper where they want to do collaborative machine learning research in biodiversity. So Google is getting in on the act, and think this is great idea. So it's a very exciting time to be a botanist. I think to be a herbariumologist. I hope I've convinced you that herbaria, and I didn't make you go through all 72, but that herbaria are a rich, rich resource. And I just want to end by thanking I have a wonderful group of people and these are the people I work with on these particular projects, but they're all awesome. And thank you very much for your attention. Thank you. Any questions?
Yeah, sorry. It sort of goes back to this question of repurposing data.
Pardon me?
Repurposing data, where you get someone who's had a particular study and collected something for a particular question, and then that's available and it's reused. And here, like the first thing that goes through my mind was when I was 19 and collecting plants, I didn't collect anything without insect damage because it wasn't pretty. So is there a time problem where the difference?
So that is a very good question. And that is exactly what Emily encountered from people saying, well, you know, botanists, they go out in the field, they collect pristine things. They like to, you know, I don't know about you, but I'm a messy collector. So I'm out there fighting the flies and I'm not really looking for the prettiest one, but I know there is a bias. And if that is the case, and she checked for that, then it would underestimate insect damage if people were favoring collecting more
But
pristine.
I'm saying at different times, it would have been
Right.
more when you were a leisurely Victorian,
Right.
as opposed to an overworked current academic.
Right. And she apparently did this statistics over the years and showed that there was no difference. But that was a valid criticism or potential criticism that people brought up. But again, it was not something to stop you from, you know, checking out that possibility. Did someone else have a question? Yes?
Has it been estimated, do you know if there is a lot of contamination of the specimen? If you want to do a DNA work, of course you can ID the species, but then some bacteria or others, spores from other plants in the neighborhood, has this been evaluated?
Oh, yes. So unfortunately, there are databases with all of the data. So what we work on a lot is chloroplast DNA. So fungal spores aren't a problem for us. Fungi don't have those. But they have been a real problem over the years. Like early on, 20 years ago, somebody published a paper about a flowering plant having these genetic alliances with some strange algae. Well, that was just because it was algae growing. It was a pond weed and it was growing with this algae on it, and they happened to sequence the algae instead of. So there was always that possibility of contamination. But over time, over these last 25, 30 years, these databases have become enormous, and so it becomes easier to identify contamination.
I want to thank you for this beautiful presentation.
Thank you.
I am in the world of mycorrhizal, not very plant, but my feeling was with the herbarium that we were at a stage of disappear of the Pryer, and people want to make it throw. But with you, it's regenerating. Yeah.
Yeah, I'm glad, because you know, it was to me when I was an undergraduate student. A herbarium was a great place to go and hang out. And sometimes curators didn't know what to do with me. And I remember one gave me a book of Agnes Chase's first book of grasses. And he said, just sit in the corner over there and work your way through all the grasses. And it was such a calming, wonderful environment. And there's so much you could learn in there. And it's so sad that it's very difficult to, and every time there's a new dean or a new administrator, we have to argue again for its value because they've never heard of them. And so that's in part why that outreach was very important to me, is to get the word out. Herbaria are great. I'd rather these kids would come and hang out in the herbarium than go around with the guns and do the terrible things. You know, if they know if people have the education or are shown for other opportunities, that's really what counts. So
The nature is there, you know. We must re-educate the children to go to nature, not only in a computer and Facebook. So we need herbarium and this kind of science. Thank you.
Thank you. Yes.
Yeah, thanks for the presentation and your introduction. I was amazed that you could regrow calypso from spores that were like hundreds of years ago. Do you have any plan to use that to study rapid evolution or what's been going on in over thousands, hundreds of years.
So that's certainly something that could be done. Finding the right student to work on that is a different thing. So it's a very, not all herbaria have, herbarium specimens have spores that are at the right condition. They vary in terms of age. And also sometimes the way they were preserved may damage the spores. But I have a tiny little group of people that I'm trying to interest in understanding this. And yes, it's important to go in the field, but you have this wealth of collections. You can make these transfers of spores, grow these up, and look for, because, you know, ferns are, because they disperse by spores, people think that they're all just long distance travelers, and they try to explain their biogeography in that way, but they have these different mating systems, and sometimes to become to be able to self, the genetic load of doing that is sometimes high, but it's as if ferns are able to play with that. A fern spore lands on an island somewhere. It's usually an outcrosser. It's able to switch to becoming an inbreeder until, you know, friends arrive to help the situation. But there are so many interesting patterns that could be looked at by growing up the spores. It's just a matter of finding the people.
Because you can get more DNA.
Pardon?
You can get a lot more DNA.
Yes, you can from these, we've had great success getting DNA from specimens that are more than 100 years old. So.
So in your presentation, you said that the herbarium is able to give like a historic bank of DNA. Do you know if there is any information that those herbarium could give, like maybe chemical diversity or?
Yeah, absolutely. So for example, at Duke, there were, Duke has also got a big herbarium, but it's got a big moss and lichen herbarium. And 50 years ago, there were a very famous couple at Duke, the Culbersons, Bill and Chiquita Culberson. They were both PhDs. Bill was a botanist, but Chiquita was a chemist and a biochemist. And so she developed a technique for extracting chemical compounds from lichens, and she published many papers. And she revolutionized lichen taxonomy by doing that kind of thing. So yes, people have used these leaves also for, I can remember we used to do isozymes before we did DNA, and we were able to get tissue that would yield isozyme data. And so that was sort of the beginning of, wow, you can't believe, you know, it's all right here, and we have it already, and we can extract it and ask a question and have an answer. It's
Did you notice if there is any difference between those chemical compounds and from the species in the past compared to the species now?
That's another great question. So there's a woman, she's an entomologist, May Berenbaum at the University of Illinois. And I saw one of her papers when I was trying to gain ammunition for herbaria. She did a wonderful study where a parsnip plant from Europe came and invaded North America like a couple of 100 years ago. And it had a pest in Europe, but the pest didn't come over with it. And so herbarium collections have been made over the past couple of 100 years of this plant. And she looked at these chemical compounds that in Europe that these plants were making. So when the plant got here, it was like it looked around and said, hey, I don't need to make those deadly coumarins or whatever they were. And so they showed a real decrease in those compounds. Then the pest did make the trip back, And almost like clockwork, the plants that had been collected post that date really zoomed up their production of these chemicals. So it's a very interesting work that people can do and have done with herbarium specimens. And another thing with this insect work is that paleobotanists have been doing this for a long time. Paleobotanists look at, I have the greatest admiration for people who can look at rocks and see the things that they see. And, but they are able to see, they're perhaps the best at identifying insect damage on leaves. And then have been able to date certain groups of insects back by many millions of years just because they found the traces of their insect damage on leaves of a particular age. Those are some of the really neat papers. You say, wow, they looked at this and oh, how neat. It's, you know, so again, those are specimens that were collected for a very different purpose and are now being utilized to do other things. You have an interest in chemistry? By any chance?
One last question.
In an era when we're losing so much diversity, or there's a potential to lose, can you see a priority or a strategy for herbariums to come to the forefront in addressing biodiversity loss?
Absolutely. There are all kinds of studies that can be done because you have this wonderful time frame and space. And so you can travel back through, so we can look back through the Anthropocene. You know, if we talk about the Anthropocene, we have a way of looking at it just through herbaria and identifying critical taxa that are in danger and how things are. We're able to make predictions also that if things continue on the trajectory that they are, for example, one little fern I described many years ago was a little oak fern from the Appalachian region, and it's endemic to the Appalachians. And I gave this project to an undergraduate student, and she did these climate models and was able to show that if we continue on the trajectory that we're at, in 50 years, the only place this fern will be able to live is Sept-Îles, Quebec. And that's not just because I would go plant it there, but it's, you know, being able to show that, that certain taxa are under great danger of being imperiled. So they do have an important place. I'd be happy to take any other questions anytime.
Okay, Kathleen's going to be here for 2 more minutes.